Okay, all right. I guess all of you have finished up watching video related to the application of DNA technology in agriculture, in medical, in what else? Animal breeding, okay? And then ethical issues or problem that may raise due to the recombinant DNA technology. So is there any problem that you would like to ask? Tak ada? Okay, so kalau tak ada soalan, I assume everyone has done your job, right? So with that, we officially finish with chapter 11, biotechnology. So today we are moving into the 12th chapter, genetic change and variation. Okay, so in this chapter of genetic change and variation, somehow it will relate chapter to chapter genetic, chapter 9. It related to chapter 10, protein synthesis, because in protein synthesis, you have learned about the process of replication, transcription, translation, right? So chapter 12 related to chapter 9 and also chapter 10. Okay, all right. So the basic knowledge is there, right, for this chapter. Okay, so in chapter 12, genetic change and variation, we are going to see how or what caused somehow the gene or somehow the chromosome can mutate it. All right, so when this thing happen, it might cause few problems, as you all already know. For example, the Down syndrome, right? Down syndrome is caused by alteration of chromosome number. So there are so many other situations caused by the change of genes, all right? Mutation of the gene and also mutation of the chromosome that cause that to happen. So we are going to learn all the factors that might all these cause this thing to happen. Okay, all right, so learning outcome. So the first learning outcome for this chapter is to explain how crossing over. So we have learned about crossing over, right? Independent assortment, homologous chromosome during meiosis, random fusion of gamete, fertilization, and mutation leads to genetic variation. So we are going to see all these factors that somehow might cause genetic variation to produce. Okay, right. So what are the source of variation? And first of all, what is variation? Okay, so variation is different in characteristic of organism that belong to the same species, meaning that a variation showing that each individual or each organism is actually different, right? Right, different due to the variation. So this variation is caused by genetic factors, right? What we call it, genetic traits ataupun what gene that you inherit from your parents and ancestors, okay, that will determine your phenotype, or we call as the appearance. Okay, second one, it might also caused by environmental factor. For example, something that influence the characteristic to develop. Let's say environment, you are grown up or you are born in an environment that very kata apa, less hygienic. Contoh kita bayangkan dekat certain part of the Africa, less hygienic, very low in terms of the medical supply, kan? Kekurangan ni lah penjagaan kesihatan dan sebagainya. So you can see that somehow all the children born in that particular area inherit certain what we call it, certain characteristics, right? Mungkin they are way too skinny, they are very light in terms of weight, right? And somehow their stomach very kata apa, resistant toward apa nama not really hygiene water, kan? So macam kita kalau pergi duduk di situ minum air diorang, kita sakit perut, tapi diorang boleh tahan. So because they have developed some kind of characteristic due to the environment factors. Okay, right. So okay, so these are other mechanisms that contribute to genetic variation. Okay, the first one, as of course we learn in genetic independent assortment, right? The second law of Mendel. Okay, and then crossing over during meiosis 1, right? So masa meiosis 1 ada crossing over. So during crossing over tu berlakunya changing of genetic material. Okay. Number 3 is random fertilization. Alright. Ataupun bahasa Melayu kita panggil sebagai persenyawaan rawak. Persenyawaan rawak, because each gamete has possibility to fuse with other gametes, right? And number 4 is mutation. So A, B, and C will reshuffle existing gene. Maknanya, for independent assortment, crossing over, random fertilization, the gene are all already present, but they are reshuffled, meaning transfer to one chromosome to another chromosome, alright? That's it, itu sahaja. But somehow, for mutation might cause the creation of new forms of gene. Kalau mutation berlaku, kadang-kadang kita akan membentuk gene yang baru. Kadang-kadang kita akan menghasilkan what we call it alteration, perubahan daripada segi kromosom dan sebagainya. Okay, so somehow when we compare A, B, C and D, so the effect of mutation lagi teruk lah. Okay, lagi kata apa, lagi besar. Okay, right. Now let's see the explanation for independent assortment. Okay, in independent assortment, number of possible combination when chromosome assort independently during meiosis is 2 to the power of n, where n is the haploid number of organism. Let's say for human, our haploid number of chromosome is 23. Untuk manusia lah, haploid kromosom kita 23. So in human, number of possible combination of maternal and paternal chromosome. So gabungan antara mak dengan ayah punya kromosom, possible combination dia can result in 8.4 millions of combination. So that is due to independent assortment. Okay, if you can't remember what is independent assortment, so remember you have homologous chromosome. All right. Okay. Okay. Let's say this chromosome carry gene capital Y, this one is small y, and this is capital G, small g. All right. Remember during meiosis, you might have one combination of gamete, yaitu you mungkin akan dapat the first combination is capital Y, and then capital G. Okay, the second combination when the meiosis takes place, you might be having combination of small y, small g, right? So first combination kita capital Y, capital G gamete, and next one is small y, small g. Okay, this is the first two combination of gamete. But when independent assortment takes place, remember, alright, chromosome ni boleh duduk sini, yang ni boleh duduk sini. So due to that, you might have another two different combination of gamete, which is capital Y, small g, and also small y, capital G. Right? So this is what we call as independent assortment. So this is only involve two chromosome homolog. Just imagine we have 23 pairs of chromosome. Right? So the possible combination of gamete is 8.4 million. Okay, right. Okay, so this is what I mentioned just now, independent assortment of homologous in meiosis. So you can see the first combination is this one, the blue, and also both red chromosomes. All right, so we have combination 1, combination 2, and somehow we might have different combination. Right, so mungkin berlaku independent assortment. Berlaku independent assortment, you can see we got this one. Okay, combination number 3 and combination number 4. Okay, right. Okay, second one, crossing over. Okay, for cross, please let me know in case you don't have, you want to copy something. So please let me know. Okay, crossing over. Crossing over produce combination of chromosome which combine genes that can be inherited from the two parents. Thus, each individual chromosome gamete in a gamete would be not exclusively maternal or paternally in origin. Okay, maksudnya apa? Remember during prophase 1 of meiosis 1. They say this is chromosome from our father and this is from the mother. Right? So what happened in the meiosis is the crossing over where, okay, boleh lukis kejap. One of the sister chromatid, right, cross. You can see they are what call it synapses one another. So the genetic content will be exchanged here. All right. So that is make each individual is different from their father and also their mother. Okay, due to the crossing over. Okay. So this is what you can see, the crossing over, right? Example, you can refer to page 316 of textbook. So due to the crossing over, so allow the homologous chromosome, recombinant chromatid, sister chromatid to separate. Okay, so ini chiasmata and cohesion between sister chromatid hold the homolog together and then move to the metaphase 1. So itulah lebih kurang explanation macam Tim cakap tadi. Okay. Okay, number 3, random fertilization. Okay, you can see as for number 1, independent assortment. Number 2, crossing over. So the genes are present, but reshuffling takes place. Okay, so similarly to what happened for random fertilization. Human ovum and sperm each represent one of the approximately 8 million possible chromosome yang kita dah bincangkan in independent assortment just now. Okay, so thus any two parent will produce a zygote with any about of 2 to the power of 23, that of 2 to the power of 23 possible combination, which is around 70 trillion possible combination. Okay, random fertilization of the gamete and also the ovum, right? Okay, and number 4, mutation. So remember for A, B, and C, random fertilization, independent assortment, and also apa tadi tu? Crossing over. All right. So the genes are already present, but the reshuffling happened. Okay. But now we are going to discuss number 4. Okay, mutation. All right. So we are going to differentiate between somatic mutation and germ cell mutation. All right. So first of all, you need to know what is germ cell mutation. Okay, somatic kita dah tahu dah. Somatic. Okay, cuba tolong Madam Zat. Find the definition of somatic mutation and germ cell mutation. Tak ada kat sini. Tak ada. Oh, ada. Malah. Baru nak suruh dia cari pagi-pagi ni. Okay. So we are going first to differentiate the somatic mutation and germ cell mutation. Okay. What is mutation? Mutation is a random change in gene which happen in gene or in chromosome which resulting a new traits. So nampak tak? So mutation can cause formation of new trait ataupun characteristic that can be inherited. Okay, mutasi akan menyebabkan berlakunya terbentuknya sifat yang baru which can be inherited. Okay, right. Mutant, right? So mutant is organism that have been changed, right? Where the gene or the chromosome has been changed by mutation. So we call as mutant. So orang ataupun organism yang mengalami mutasi, we call it as mutant. Okay. All right. So let's look at somatic mutation first. Somatic mutation happen in somatic cell. Okay. Somatic cell apa? Somatic cell are all cells.
Yes, selain gamete.
Yes, except for gamete cell. Ataupun reproductive cell ataupun cell that reproduce gamete. Okay, somatic cell are all cell except for gamete cell ataupun cell that produce gamete, referring to the ovum and referring to the ovary and also the testis, which produce sperm and also ovum. Cell that produce gamete, yaitu sperm and also ovum. Okay, alright. So mutation of somatic cell can be inherited to the daughter cell by mitosis. Maksudnya somatic cell boleh diwarisi, but it happen by mitosis. Okay, it is not transmitted to offspring. Maknanya dia tak akan kata apa, terjangkit ataupun dipindahkan kepada offspring, anak-anak. Kalau berlaku somatic mutation. Okay, let me just give you an example. Pernah tengok tak orang yang ada pipi dia macam besar kat sini tu? Alright, so that is somatic mutation. Maksudnya mutation happen to the tissue dekat dia punya pipi ni, dekat dia punya rahang bawah dengan rahang atas ni. So dia macam bengkak kat situ kan, macam beguk, orang panggil beguk tu. Right? So that is somatic mutation. So if he has this somatic mutation will not be inherited kepada anak-anak dia. So anak dia tak adalah kena, because it happened to his somatic cell only. Okay, alright, so tu. Okay, it may give to uncontrolled growth or mass of cell, which we call as tumor. So somatic cell mutation can cause tumor, right? The tumor can be either benign or malignant. Okay, malignant means apa? What is benign means?
Bahaya.
Malignant maksudnya apa? Dia boleh apa?
Boleh cancer.
It is, yeah, it can cause cancer. Cancerous lah. Alright. Malignant means it is able to spread. Okay, dia boleh merebak. That is malignant. Contoh kalau start dengan the tumor happen dekat pipi contohnya. So it can spread to your tongue, it can spread to your eyes, for example. Okay, right. Kalau benign means it is not cancerous. Contoh macam orang yang ada pipi besar tu, benguk pula dah. Beguk tu, alright. So that one, that is benign. Usually it is benign. Okay, so it is not cancerous lah. Not cancerous and it is not able to spread. Dia duduk situ je. Dia tak ada nanti merebak habis hidung dia. So jadilah macam yang cerita putah tu. Okay. Right, second, germ cell mutation. Okay, germ cell mutation is mutation that happen in the gamete or in the cell that give rise to gamete. So cell that give rise to gamete, we can refer to testis, right? Or cell of the testis or cells of the ovary, because these cells give rise to the gamete. Okay, so itulah, right? So germ cell mutation is the mutation that happen in the gamete. Okay, so it occur by meiosis in reproductive tissue or in the germinal tissue. Okay, and it can change genome of the gamete. Remember, genome is the total DNA. Genome ialah keseluruhan DNA. So it can cause change of the DNA, the total DNA of the gamete, right? And it can be transmitted to the offspring and also future generation. Okay, so this is the difference between germ cell mutation and also somatic mutation. Okay. All right. Now let's see what factors. Yeah, please let me know if you want to jot something. Okay. Okay. List down the factors that cause mutation. So what cause mutation to take place? So kita dah tengok effect dia. So mutation ada dua, somatic mutation or germ cell mutation. So somatic mutation, yeah, somehow it cannot be transmitted to the offspring. Germ cell mutation can be transmitted to the future generation. So now let's see what caused the mutation. Okay, so some of you might be further in radiotherapy, for example, because some of you were allied health science kan, student. So allied health dah boleh pergi radiotherapy. So you need to know lah all these things, right? Okay, cause of mutation. Okay, so mutation happen due to error during DNA replication, during DNA repair, or during recombinant. DNA recombinant. And it is what we call as spontaneous mutation. Remember, DNA replication happen during S phase. Kan? S phase of interphase. All right. So somehow there are errors happen during the DNA replication or during the DNA repair within the cell cycle. Remember dekat cell cycle ada checkpoint kan? Kalau you ada terbaca bahagian checkpoint tu, right? So when the DNA repair happen, somehow the checkpoint is not able to detect changes happen to the or mutation happen to the chromosome. So that's why it happened lah. Okay, right. Second one is induced by mutation. So what is mutation? Mutation is agent that can cause mutation. Okay, what are the mutation? They can be physical mutation like high energy radiation from X-ray ke, from UV light ke, right? That can cause the DNA to break up. So in other words, DNA break up ni maksudnya DNA kita damage ataupun dia jadi single strand. The double stranded separate into single strand. So this is the effect of radiation. Okay, that's why for person who is in the radiography area, right? So whenever they want to ataupun sesiapa sahaja yang nak pergi X-ray kan kena pakai protective vest, right? Protective cover. So that cover is actually helps to filter the radiation. Supaya tak kena radiasi yang kuat lah pada orang. So that is why. Okay, right. And if you look at cerita Fantastic Four, for example, kan? So macam mana diorang all the 4 scientists tu, scientist ke 4 tu? Tak ingat. Okay, all the 4 person tu suddenly become somebody else because they have been exposed to very high radiation dekat space kan masa tu. Kalau you tengok Fantastic Four. So that is the effect of X-ray radiation towards DNA. Okay. All right. So this is what you can see. You can X-ray or a gamma ray, whatever. It can break the DNA, and you can see it can cause the DNA strand to be exposed. So when the DNA strand is exposed, something else can happen, which we are going to learn after this. So bila DNA ni dah terdedah, so benda-benda lain boleh masuk antara nucleotide sequence of the gene kat situ. Okay. Alright, second one is chemical mutation. So chemical mutation are agent cause mutation that came from chemicals. Example, 5-bromodeoxyuridine. Okay, ni kena hafal lah nama dia kan. Or we call as 5-BU. Okay, so this 5-BU mimic thymine and cytosine. So maksudnya rupa, in other words, I would say bentuk 5-BU ni rupa dia nak sama dengan thymine and cytosine. Okay, nak sama dengan T and also C. So what happened? They resemble the normal bases in DNA and cause incorrect base pairing during replication. Okay, Mina bagi contoh lah. Let's say 5', 3'. Okay, this is our normal DNA. Okay, sepatutnya this is our double-stranded DNA. Okay, sepatutnya kalau our nitrogenous base is T, it is complementary to A. If it is C, it is complementary to G. Right. So if mutation takes place with this chemical 5-BU, okay, so what happened? It might make thymine or it might make cytosine. So let's say sini bukan T dah, B. Okay, the 5-BU, B. So it complementary to A because it is the 5-BU mimicking thymine and mimicking cytosine. So what happened? The base pairing will be wrongly paired. Alright, so yang ni tadi betul lah, C, G. So what happened? The sequence will be changed. Nucleotide sequence tu berubah. So what happened? It might not produce the specific ataupun produk yang sepatutnya. So dia akan membentuk benda yang lain. Itu right. So itu kesan chemical mutation. Okay. Second one, instead of 5-bromodeoxyuridine, so we have another one which is ethidium bromide. So ethidium bromide cause frame shift mutation. So what is frame shift mutation? Kita akan belajar kejap lagi. The idea is tak apalah, kejap lagi kita belajar. Okay, frame shift mutation. Okay. And the third one example is nitrite preservative. Bahan pengawet yang mempunyai nitrite. Bahan pengawet And what happened? Nitrite preservative substitute structure of bases and interrupt the process of replication. So dia akan menukarkan nitrogenous base. Tukar, substitute Dia akan tukarkan base. Nanti kita akan tengok contoh-contoh lepas ni, right? And it will interrupt the process of replication. Okay, nitrite preservative banyak ada dalam makanan. Alright, contohnya macam ni lah. Sodium nitrite. Ni. Right, so it is additive lah. Additive that is added into meat to prevent the meat from spoiling easily. Okay, supaya daging tu cepat rosak, dia letakkan this preservative tu untuk mengelakkan daging rosak. So what happen is nitrite preservative is carcinogenic. Carcinogenic means okay, can cause cancer. Okay, so tu. Alright, so other mutation, right, damage the DNA structure. But most physical and chemical mutation that we were discussed just now are carcinogenic. Carcinogenic mean they can cause cancer. Okay, right. So tu. Okay, so far any question? Okay. Okay. Now let's go to the further discussion about the mutation. Okay, all right. Describe different type of gene mutation. Okay. Now, first we are going to discuss on gene mutation with examples and consequences. All right. So they are substitution. Right. Example is sickle cell anemia. Insertion or addition. Example is Tay-Sachs disease. Okay. Deletion. Example cystic fibrosis and also thalassemia major. And we are going to differentiate what is missense, nonsense, silent mutation, and frameshift mutation. So semua ada yang dalam LO ni semua penting. Okay, right. Okay, now let's see first types of mutation. There are two types of mutation. Generally, there are two types of mutation. The first one is gene mutation. The second one is chromosomal mutation. Okay, what is gene mutation? Gene mutation is alteration in the sequence of nucleotides in the DNA. So let me give you an example. Okay, let's say this is our DNA. Okay, so sequence to, let's say lah, sequence untuk menghasilkan insulin. Okay, sequence of gene, sequence of DNA to produce insulin is, let's say A, T, T, C, G, C, A, G, for example. So this is the nucleotide sequence specifically, containing gene that is code for insulin. Contohnya, inilah dia punya sequence untuk menghasilkan insulin. So what happened? This sequence is altered. A, T, T, G, A, C, G ni is altered. Altered may be due to deletion. Contoh, what is deletion contohnya? Contoh kalau deletion. Contohlah, G ni hilang kat belakang tu. Nucleotide sequence G tu dah hilang. So bila hilang, now you can see the sequence become ATTGCA. So ATTGCA cannot produce insulin because the sequence has been changed. So this is what we call as gene mutation. Okay, alright. So macam tu lah, itu antara satu contoh dia. So gene mutation is alteration in the sequence of nucleotide of the DNA. Right. As for chromosomal mutation change in the number of chromosome or change in the structure of chromosome. Okay, what does it mean by change in structure? Okay, let me give you an example. Okay, let's say this is homologous chromosome. Sepatutnya setiap kromosom kena homolog sebab one chromosome datang daripada ayah, satu kromosom datang daripada mak. So let's say this is chromosome number 14. Contohnya, kromosom nombor 14. So what happened? Change in the structure of chromosome. Salah satu sisterchromatic ni berubah. Pendek sebelah je. Nampak tak? So change of the structure of chromosome. So when this structure of chromosome change, it can cause changes of the gene. Alright, banyak gene akan berubah. Because remember, setiap kromosom ni ada locus. So many locus. You just imagine satu ini je hilang, dah tak ada. So it will affect few genes. Okay, so that one. Okay, what about changes in chromosome number? Okay, let's say for human, kita ni biasa ni our 2n diploid number chromosome 2n equals to 46. because kita datang 23 plus 23 kan. 23 from the father, 23 from the mother. So total 46. So there is a what you call it, condition where chromosome mutated in terms of chromosome number. Daripada 46 tiba-tiba jadi tambah 1. Jadi 47. So jadilah dia Down syndrome. Macam tu. Okay, alright. So that is one example, right? Okay. So we are going to discuss first with gene mutation. Okay, another name of gene mutation is point mutation. Point, meaning it happened at specific nucleotide sequence. Okay. So the gene mutation involve deletion, insertion, and substitution. Substitute, bertukar. Insertion, maksudnya addition. Okay, right. So gene or point mutation is alteration in the sequence of nucleotide in the DNA, right? The change can affect single nucleotide pair, or it can affect larger gene segment of a chromosome. So effect dia berlaku dekat single pair of nucleotide ataupun larger gene segment. Okay. And the changes will be transmitted to mRNA. Sebab kalau DNA tersilap, berlaku mutation of the DNA, it can somehow affect the mRNA during transcription. So when mRNA change or affected, somehow it can change the amino acid during translation. Okay, so itu kesan dia. So berlaku changes of DNA, it will affect the mRNA during transcription. So mRNA you berubah, of course the amino acid also will be changes. Okay. Okay. Let's start with point mutation. Ingat tadi mutation ada dua, gene mutation atau nama lainnya point mutation, and yang kedua nama dia chromosomal mutation. Right. So point mutation or gene mutation is the most common type of mutation. Yang lebih banyak berlaku ialah dalam bentuk gene mutation. Okay. It changes a single nucleotide base pair. Alright. And that is due to substitution, insertion, or deletion. Okay. Ini sebab-sebab dia yang menyebabkan point mutation happen. Okay. Let's discuss substitution dulu. Am I too fast?
Okay, boleh.
Boleh. Okay. Right.
Boleh.
Okay. Substitution. Okay, kita start dengan substitution. Okay, kat sini Melinda akan slow sikit dah. Right. Substitution is replacement of one nucleotide and its partner with another pair of nucleotide. Substitute, bertukar. Right. So when one nucleotide change, it will also change the complementary base pair. Okay. Right. So itu kesan substitution. Kalau one single strand punya nucleotide berubah, dia ada strand also akan berubah. And the effect can be either silent mutation, missense mutation, or non-sense mutation. So these are the effects of substitution. Okay, kita akan tengok satu-satu. Contoh silent, contoh missense, and also contoh non-sense. Okay. Alright. Let's start with silent mutation dulu. Okay, ingat this is due to substitution. Okay. Betul kan substitution? Yes. Okay. Nucleotide pair substitution result in codon coding for the same amino acid. So this is what we call as silent. Maknanya apa? Berlaku mutasi. Berlaku mutasi. Yes. The nucleotide and its base pairing has changed, right? Due to substitution, bertukar. So bila one nucleotide bertukar, its partner or its complementary base pair also bertukar. Tapi pertukaran tu menyebabkan it still produce the same amino acid. It still produce the codon that code for the same amino acid. So bila benda ni berlaku, it called as silent. Maknanya takkan ada kesan yang terlihat. You cannot see any different because mutation happen, but it does not showing you obvious effect, which is it does not change the protein, it does not change the amino acid. Okay, that is what meant by silent mutation. Alright, okay. Kita tengok contoh. Okay, tolong fokus untuk setiap contoh. Okay, if let's say given to you this kind of question in exam, so usually it will give you the DNA strands, the mRNA and whatnot. Okay, now look at here. This is your DNA in normal condition. Okay, this is your DNA strand given to you A, B. Alright, ni sambil-sambil ulang kaji ya, sambil-sambil ulang kaji chapter 10. Okay, alright. So this is your DNA. Now, tell me, which strand of your DNA that will be used during transcription of mRNA? DNA strand A or B? Which one is the template strand yang akan digunakan? mRNA kita akan start dengan 5. Which one that will be used as template?
A.
A. Very good. A. Ingat because our mRNA start with 5' sebelah kiri. So the DNA strand that will be used as template is A, because you can see it start with 3' from the left. So it must be anti-parallel. Ingat tu. Tolong ingat. Okay. So this is our template strand. Okay, all right. So remember how to do it, how to make your mRNA. If your DNA is T, your mRNA will start with your mRNA ya kan? mRNA awak apa? T?
E.
Apa dia? Again?
A.
A, very good. Okay, next. A.
U.
A of DNA, U, very good. C.
E.
Teruskan.
A.
Mhm.
A.
Mhm.
T.
Okay, good. Teruskan.
U.
Yes, very good. Lagi. Mhm. 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 Mhm.
U. A. A.
Allah. Okay, that is your mRNA. So remember, every 3 consecutive, for every 3 consecutive mRNA base, we call it as 1 codon. Okay, so we have berapa codon kat sini? Okay, ulang kaji sambil-sambil buat latihan ni. Berapa codon ada?
5.
5. Kita check betul tak? 2, 3, 4, 5. Jawapan dia salah. Jawapan dia bukan 5. Not 5. Soalan suka tanya ni, soalan suka tanya benda ni. Why there is only 4 codons?
Because the last is the stop codon.
Very good. Okay, soalan akan tanya you, what is the total number of codon? Okay, the answer is 4. Explain your answer. The 5th codon is stop codon. Okay, stop codon does not code for any amino acid. Alright, and there is no tRNA for stop codon. Okay, faham ke yang tu? Okay, alright, ulang kaji. Sambung, ulang kaji sebelum Madam tekan slide ni. Okay, now from mRNA, next the proceed after that is the translation. Okay, so during translation, each of the codon will be translated into amino acid, specific amino acid. Okay, so now What is the amino acid code by codon AUG? Okay, please open your genetic table. Kalau rasa tak ada nota tu, Google genetic table. Sebab ni sambil-sambil ulang kaji ni. You dah faham yang ni, insyaAllah yang lain you faham.
MET.
Yes, MET. Medan dah suruh hafal kan? AUG is start codon. So start codon memang code for only MET. Okay, next one.
LYS.
LYS eh. LYS. U.
PHE.
PHE. Remember the first letter must be capital. Okay, lepas tu.
LY.
Apa?
GLY.
Oh, GLY. Okay. GLY. Okay, nampak? So stop codon does not code for any amino acid. So this is your product of normal transcription and translation of DNA. Okay, right? Faham yang ni? So what if? Okay, so kena hafal MET, LYS, codon-codon tu tengok. Okay, this is wild type. Okay, wild type means normal Wild type means normal. Normal DNA without mutation. Okay, so you have look at here the codon. Okay, kita dah tengok dah codon dia, and then we have look at the protein, right? We have look at the chains of the polypeptide from the amino acids too. Okay, right. So now, what if mutation happen? Okay, jap. Kita padam dulu yang lain-lain ni. Okay. Now, what happen when silent mutation takes place? Okay, please note. Oh, jap. Note note this one. Tengok yang kat sini. Okay. Nucleotide G, right? And its base pair has been substituted. Okay. Now G and its complementary base pair C has been substituted into A. So bila A berubah, maka the what you call it, nitrogenous base pairing also will be changed. Okay, so now you can see tadi apa? G change, substitute substitute into A. Okay, so bila dia dah berubah jadi A, so what happened to your mRNA? Just now C. So now dia akan tukar jadi? Tukar jadi apa? Ingat nak ikut yang mana ni?
Jadi U.
Apa dia?
U.
U, yes. Sebab remember we have to follow the template strand. So A will be changed into U. So look at here. GGU. Look at the genetic table. GGU code for which amino acid? GGU code for which amino acid? Tadi GGC code for glycine. Now GGU code for?
Also glycine.
Also glycine. Nampak tak? Alright. So it's still producing glycine. So mutation happen due to substitution, but it still producing or coding, producing codon that code for the same amino acid. So it's still producing glycine. So this is what we call as silent. Maknanya produk kita sama walaupun berlakunya mutation. Nampak tak? Right? So there is no observable effect of mutation can be seen. Okay? So itu kita panggil silent. Okay, any question? Faham ke tak silent tu?
Faham, Madam.
Okay.
Jadi, Madam, soalan. Silent mutation, is it dangerous or not since it doesn't change the product of the amino acid?
Semula?
is silent mutation dangerous or not?
Oh,
Sebab
in
dia-
this case kita panggil tak dangerous lah. Because it does not change the product. It does not change the polypeptide or amino acid tu. Okay, dalam kes ni kita panggil tak lah. Tak bahaya. Okay, sebab dia tak mengubah apa-apa. Macam tu lah. Okay. Alright, lagi soalan? Faham Habis tu nanti medium soalan exam keluar macam mana nanti. So dia akan bagi you this kind of mutation, for example, and dia bagi you normal, dia bagi you the mutation, and then dia bagilah situasi, look at the protein and whatnot. If you look at the protein producing the same amino acid, so the mutation that happened is silent mutation. Silent mutation is a kind of gene mutation, sejenis gene mutation, tapi jenis yang mana? Jenis yang silent. So you have to look at from the top sampai bawah, tengok apa yang berubah and adakah ada benda yang berubah dan sebagainya. Macam tu. Okay? Right. Second one, missense mutation. Missense. Okay. So missense mutation is nucleotide pair substitution, still under substitution. Nucleotide pair substitution result in coding for different amino acid. So you can see this kind of mutation producing different codon, and this codon produce or code for different amino acid. But missense mutation can have little effect on protein produced because, right, maksudnya dia memberi kesan kepada protein yang akan terhasil, tapi kesan dia sikit je lah, taklah efek sangat. Okay, a little effect on the protein produced because the amino acid may be changed to another amino acid, but with the similar properties. Faham tak maksud dia? Maksud dia ialah amino acid jadi lain. Amino acid change. Alright, but the amino acid have same property. Sifat dia sama. Remember, we dah belajar chapter 2, property of amino acid. Some of the amino acid is acidic. Some amino acid is basic. Some amino acid is polar. Some amino acid is non-polar. So, in missense mutation, the amino acid change, right? But having the same property. Ingat, kan dalam acidic pun ada berapa jenis, dalam basic pun ada berapa jenis, so they are still having the same property, even though they are changed into different one. Okay? And next one is little effect on protein produced because the amino acid, the change of amino acid occur in region where the exact amino acid sequence is not functional. Okay, mungkin mutation tu berlaku pada DNA dekat tempat yang exon. Alright, dekat region of exon. So bila exon, remember during RNA splicing, it will be removed. So it does not affect much to the what we call it, to the protein, ataupun it happen to region where the amino acid produced is not essential, bukan jenis amino acid yang perlu. Essential amino acid kan, tu. Ataupun memang tak perlu nak function pun, kawasan tu tak terlibat. You bayangkan exon je lah, maksudnya the changes happen at the exon region, so it is non-functional. Okay, so effect will be very little. Okay. Okay, example. Okay, this is the wild type. Okay, remember this is our template strand. Now let's see the changes happen where. Okay, the substitution happen here. Okay, C has been substituted to T. So the complementary base pair of the DNA also change. So you can see when T of the DNA is substituted, so during transcription, T is going to be transcribed into A of mRNA. Okay, so A of mRNA, then you can see the codon change. Asalnya codon dia sepatutnya apa ya? Sepatutnya codon dia adalah GGC. Sepatutnya kan? GGC kita dah tengok tadi, it should be code for apa ni? Tak nampak lah, hilang. GGC sepatutnya code for GLY lupa lah. GLY ke? GLY. Okay, sepatutnya amino acid dia GLY. But now, due to this mutation, you can see it changed into AGC. So AGC is what I call it, substitute into serine, SER. So serine and glycine is different amino acid, but it is within the same property. Macam tu. Tahu? Okay, so itulah maksud dia. Okay, any question? Any question? Faham? Okay. Other effect of substitution. Okay. Selain daripada tu, apa lagi effect substitution? It can also affect protein function, which is if the changes occurs in the active site. So substitution also boleh menyebabkan protein function tu berubah. For example, if it happen within the active site. So kalau active site berubah, it can affect the protein lah as a whole. Okay. And sometimes substitution can produce protein with improved capabilities. Kadang-kadang mutation ni ada hikmah dia lah, which is it can improve proteins. Okay. Satu. Alright. Example of substitution. Substitution. Okay, what is the example of substitution? The example is substitution. All right, is sickle cell disease. Okay, sickle cell disease. Okay, now sickle cell happen when single nucleotide from T. Ni kena hafal sickle cell dia apa yang berubah? Okay, sickle cell happen when single nucleotide T is changed into A in the DNA template that leads to production of abnormal hemoglobin. So you can just imagine only the cause of change or substitution of one nucleotide sahaja, it caused the amino acid, it caused the hemoglobin to change its shape, right? Berubah terus bentuk hemoglobin. Okay. So this is normal, what you call it? Normal genotype, normal allele, allele for normal hemoglobin HbA, and this is HbS is allele for mutant hemoglobin. So maksudnya kalau orang tu ada hemoglobin, orang tu ada sickle cell, so the allele is HbS. Okay, right? So tu. Alright, HbS, right? The sickle cell will cause the lower amount of oxygen transported. So disebabkan oleh bentuk hemoglobin itu berubah, okay, jadi sickle macam ni, nampak? Hemoglobin itu jadi bentuk macam ni. So it somehow affect the amount of oxygen that can be carried by the hemoglobin. Okay, and number 2, it also, okay ni. The amino acid change when the nucleotide sequence change, right? It also can change the mRNA, right? So when mRNA is changed, it somehow can change the amino acid during translation. Amino acid change from GLU to VAL. So this can alter the structure of hemoglobin and cause it to form insoluble fibrous strand, so that it cannot carry oxygen effectively. Sebab apa? Kita dah belajar chapter 2, protein. Kalau insoluble, meaning it cannot dissolve in water. Maksudnya dia tak terlalu telap dengan air. Sedangkan hemoglobin kita running along our blood, within our blood, right? So that's why it will, what we call it, it will affect, or it will somehow, affect the amount of oxygen carried by the hemoglobin. Okay, right. Sickle cell also rupture easily and cause hemolytic anemia. So this kind of hemoglobin boleh pecah dengan senang, right? So boleh menyebabkan orang tu kekurangan sel darah merah. Okay, ni anemia. Alright, and also since the shape is sickle like this, right? Bentuk dia jadi bentuk sabit, so it can block capillaries. Sebab dia bentuk dia sabit, so dia boleh melekat dekat dinding-dinding blood capillary kita, and somehow it can interfere the blood flow. Okay, so let's see this one. So this is sickle cell hemoglobin. Okay, you can see here, this is the normal hemoglobin sequence. You can see T, is changed into A of the template strand, okay? So T is substituted into A. So what happened? The nitrogenous base pair also will be changed, and thus change the codon, right? Instead of ni, dia jadi ni. And when codon change during transcription, so it also might cause the changes of the amino acid. Okay, so then you can see what happened to the hemoglobin lah, kan? Right? Okay, any questions so far? Tak ada. Okay, kalau tak ada, next we go to nonsense. So kita dah discuss silent mutation, kita dah discuss missense mutation. Remember missense, it changed the amino acid, but the amino acid can have still having the same property. Okay, right. Nonsense. Okay, what is nonsense mutation? Nonsense mutation is the amino acid codon change into stop codon. Bukan jadi amino acid lain dah, terus jadi, bukan jadi codon lain, terus jadi stop codon. So what will happen? It will form shorter non-functional protein. You just imagine, sepatutnya the sequence should be this length. Now they become shorter because of the stop codon. Okay, so then it become non-functional protein lah. Okay, contoh. Contoh, this is our template strand of normal DNA. Now let's see, okay, the mutation happened here. Okay, so T is substituted into A in this case. Nampak? Tukar kepada A, right? So A of the DNA will be transcribed into U of the mRNA. So just now sepatutnya kita punya mRNA lagi panjang. Now you can see the sequence become UAG dekat sini. MET UAG. So UAG is stop codon. So bila stop codon, the rest will be non-functional lah langsung. Okay. So our amino acid is only MET sahaja, right? So this is what we call as nonsense mutation. Okay. Right. Habis substitution. Any question on substitution and the effects? Remember? 3 effects: silent, nonsense, missense. Is there any question about substitution before we move on with insertion?
No,
Tak ada? Yes. Okay. Alright. So kalau tak ada soalan, so let's have 5 minutes break. Alright. Before we go to insertion. Okay. Alright. So take 5 minutes break. Take 5, eh? Okay, let's continue. Right, second type of point mutation or gene mutation is insertion. Okay, if substitution, just now what we have discussed earlier, substitution is when one nucleotide is substitute with another nucleotide. All right. As for insertion, insertion is addition of nucleotide pairs in a gene. So what happened? There is external DNA nucleotide that is added into the sequence. Okay, maksudnya ada benda baru masuk dalam sequence tu. Contoh kalau sequence tu ialah, let's say the sequence is ATCG, for example, right? So there is additional sequence added in between here, T and C. Example, A. All right. So what happened now? Your sequence changed into ATACG. All right. So this is what we call as this is what we call as insertion. Okay. So insertion produce new sequence of codon at and after the point of mutation. Right. So it will change the codon from the start of the insertion and to the what we call it following or after the point of mutation. Okay. Right. This is what we call as frame shift mutation. Okay, so frame shift mutation is alteration of triplet grouping of the nucleotides. Okay, kita bagi contoh. Okay, contoh sebelum tak ada benda ni tadi, sepatutnya kita punya triplet set is A, T, C. This is one set, one triplet of nucleotide. But now, bila kita dah masukkan tadi, kat sini kita masukkan A contohnya. So, what happened? Our nucleotide triplet is changed. Daripada ATC now dia jadi ATA. So it will change all the what we call it triplet grouping of nucleotide. So this is what we call as frame shift mutation. Okay, right. So let's see. Frame shift mutation cause extensive missense. Remember, missense macam apa kita nak fahamkan missense tadi? Kesalahan. Tu kan missense, termiss. For substitution, dia mungkin akan termiskan one codon, berubah and change into different codon, but still code for this code for the similar property of amino acid. But for insertion, it can cause extensive missense. Kesilapan tu akan lebih banyak, right? Extensive missense, ending sooner, or premature termination, or can also become nonsense. So ini kesan kepada addition due to the frame shift mutation. Okay. All amino acid at and after the point of mutation will be changed. All right. So protein produced will be non-functional unless protein yang akan terhasil will be non-functional unless the frame shift occur near the end of the gene or the frame shift occur in multiple of 3. Okay, we are going to see each of the example. Okay, right. Maknanya apa? Kalau insertion tu berlaku 3, nampak tak tangan medium? Okay, kalau insertion tu berlaku 3, maksudnya 3 nucleotide masuk sekaligus. So dia tak akan memberi kesan yang agak banyak lah. It only change one nucleotide. It will change only one codon. Kan? Okay. Okay, let's see this one. Contoh dia. Okay, look at here. This is the wild type, normal. This is our template strand. All right. So let's see now what happened. Insertion happened here. Dekat sini. Between T and T. Okay. Cuba kita buat sekarang ni. So our mRNA will start with 5 prime. So our mRNA will be T. Jom. What? T. A.
E. U.
A. U.
G.
G. And then?
E.
A. And then?
Oh,
Apa
ada C.
bila C dah masuk, dia akan jadi?
G.
G. Because insertion happened here between T and T. So C is now here. So what happened? The next T will be shifted like that. So this is what we call as frame shift mutation. Okay, so next is, here is, so you can see starting from the point of mutation to and the following sequence will be changed. So this is what we call as frame shift mutation. Okay, so ni T, lepas tu ni G, lepas tu U, lepas tu G, C, and then U, A. Now look at the triplet codon. Okay, look at the amino acid. Okay, this is one codon, right? This one will be one codon. Nampak tak? Right? This is will be one codon. This is one codon. This is one codon. Nampak tak? Due to the frame shift. Due to the insertion. So where the effect of insertion is, it can cause frame shift mutation, and yet it is extensive missense. Remember what is extensive missense just now? Extensive missense is, apa dia cakap tadi? Extensive missense mean it can cause the changes to be more extensive, berterusan, semua berubah. Starting from the point of mutation, dia akan berubah. So this is what we call as extensive missense. Okay. All right. So let's see the effect. Okay. So what will happen? Okay, C dah masuk tadi kan? Okay, nampak tak? Tu. So all the mutant DNA will be changed. Macam mana yang kita buat tadi ni lah. Right? So change, frame shift mutation takes place, and it also change the amino acid. Okay, AG. Macam ada salah je ni. Tengok sekejap. AUG.
Yang T tu madam.
Mana?
AGT.
AGT. Dia sepatutnya
AGA.
AGA. Kan nampak tak? Benda tu lah selalunya student dah kata silap macam tu. Nampak madam boleh silap kan? So kita kena hati-hati bila buat benda ni. Okay, padam balik. Kenapa tak cakap dari tadi? Penat tulis panjang ni. Okay, apa? Nampak tak? T, A, and then next due to the insertion of C, dia jadi G, and then C, tadi apa kat sini? T T, A, and so on. Nampak tak tu? Okay, right? So as the mRNA sequence change, so it might also change the amino acid. Nampak tak? All the amino acid change. Tadi sepatutnya ni, now it change to this one. Alright, change to different amino acid. So this is extensive missense. Semua dah salah. Starting from the point of mutation. Semua berubah. Right. Second one, selain daripada extensive missense, it also can cause premature termination due to the insertion. Okay, let's see. Okay, this is our normal strand, and this is a template strand. Okay, insertion of A, right, happen between C and T, right? So frame shift mutation takes place. Okay, you can see frame shift mutation happen, right? Starting from the point of mutation, and thus it also changed the mRNA. So what happened due to the insertion, you can see that there is premature termination where the codon change into stop codon. Okay, right? So what happened to your protein? Now your protein become non-functional. Okay, right. Okay, example. Okay, sekarang ni kita cuba buat satu condition. Okay, let us do one condition. Yang ni tadi. The protein produced are non-functional unless the frame shift occur near the end of the gene or in multiple of 3. Okay, what does that mean? Okay, kita cuba buat sendiri. Kita buat kat sini. Okay, kita cuba buat Alright, what does that mean by in multiple of 3? So kalau in multiple of 3, meaning, let's say for example, okay, the what call it, insertion happen in multiple of 3 nucleotide. let's say masuknya A, T, C. Nampak tak? 3 nucleotide is added or inserted. Alright? So what happened? You punya mutant DNA, your mutant DNA akan jadi yang depan ni semua betul. TAC, TTC ni semua betul. AAA ni semua betul. CCG ni sama. And now we have insertion of 3 nucleotides. A, T, C. And this is A, T, T. Nampak tak? Alright, so bila berlaku in multiple of 3, you can see the effect is not that much. Kan? Alright, so yang ni akan sama. Your mRNA, T, A, U, G, A, G, U, G, G, C. Now, this is the mutant just now. Kan? U, A, G. This one is U, A, A. 5 prime, 3 prime. Okay, right. So nampak tak? So when your mRNA change by insertion of multiple of 3, you can see effect dia ini masih sama, ni sama, ni sama, ni sama. Yang akan berubah kat sini saja. Cuma kebetulan ini adalah stop codon. Dia bertukar kepada stop codon. Right. Cuba kalau kita ubah yang bukan stop codon, you can see it change into different amino acid. Right? Faham ke tak tu? Or let's say it happen in the end of the gene. Nampak tak? Dia duduk dekat hujung-hujung. So yang depan ni masih lagi sama. So the effect is not that much lah. It is not extensive missense. Okay? Boleh faham ke?
Faham.
Okay, right. Let's see the example of disease due to the insertion. Okay, Tay-Sachs. This is happened due to insertion. Tay-Sachs disease. Okay, what is Tay-Sachs disease? Tay-Sachs disease, all right, is an autosomal recessive disorder. Autosomal. It involves chromosome number 1 and chromosome until chromosome 22, right? So it is autosomal recessive genetic disorder. It is caused by accumulation of harmful quantities of ganglioside in the brain and the nerve tissue. Maknanya apa? So this Tay-Sachs is caused by accumulation of ganglioside. Ganglioside is good, but somehow it cannot be accumulated very, very long and accumulated without being processed in our brain. So gangliosida, bahasa Melayu gangliosida kan, kalau kita tengok iklan susu kan, mempunyai gangliosida yang tinggi, membantu sel-sel otak berhubung dan lain-lain kan, right? But this quantity of harmful ganglioside is not good for the brain and also nerve. Okay, so dia akan menyebabkan Tay-Sachs ni lah. Right. So you look at the template strand of the DNA. So it is happened due to insertion of 4 nucleotides, A, C, A, T. Right? Masuk dekat dalam within the template strand and it will cause, bila masuk, apa kita panggil tu? Cause what? It cause, bila something masuk, yang lain berubah, starting from the point of insertion onwards, kita panggil apa tu?
Extensive missense.
Sebelum terjadi extensive missense, dia adalah kita panggil apa ni?
Frame shift mutation.
Yes, frame shift mutation dulu. Right, so the insertion caused frame shift mutation, which the effect is, you can see it will change the, what we call it? Nucleotides, right? And it base pair, it will change the mRNA sequence. So ini kita panggil sebagai, it will cause extensive missense. Okay, macam tu. Faham eh? Alright, so efek kesan sepenuhnya adalah extensive missense. So you can see what happened, right? So should be your normal protein, but due to the insertion, it causes different production of protein. So this TSH is due to deficiency of enzyme beta hexoaminidase A, right? The insertion happened in 4 base pair in exon number 11 of the hexoaminidase gene that result in the frame shift mutation. Okay, so insertion tu berlaku dekat exon number 11 dan dia akan affect lah the whole sequence. Okay, alright. So and thus resulting the frame shift to happen. So the person with TSH dia tak ada enzim ni. Beta hexoaminidase A, dia tak ada. So bila tak ada this hexoaminidase A, they cannot metabolize, right, the excess ganglioside that accumulate in the brain and also nerve tissue. Okay. Now, soalan ni ialah what will happen to the person with TSH? Okay, right. Look at here. Nampak ni? Due to the insertion, you can see that the mutant protein produced are this instead of this. Nampak tak berubah starting from the point of mutation onwards. Okay, right. So this is what happened to TSH disease. Right. So person with extensive with TSH disease will have, kat sini ada ke tak? Oh, tak ada. Sini ada. You ataupun you boleh baca. You can see it cannot break the fatty acid in the body, and because of this there is a build of fatty substances such as an external that they reach to toxic level in the brain, where the child start to be affecting the nerve cell and causing variety of symptoms like blindness, and eventually can lead to death. Right, itu kesan TSH. Sebab dia boleh efek mata, sebab dekat otak kan, boleh efek mata dan boleh menyebabkan kematian. Kepala baby tu besar, right? That is also one symptom ataupun one characteristic of TSH. Okay, you can read further. Nanti Google je TSH, okay? Right. Any question? Okay. Next, yeah.
Yang extensive missions tu, dia punya stop codon dia kat mana?
Stop codon dia kat mana? Mungkin lagi hujung lah. Mungkin lebih-lebih ke belakang lagi. Tapi kita tak tunjuk lah. Kita nak tunjuk yang berlakunya perubahan starting from the point of mutation to sampai habis. Macam tu.
Okay.
The aim ataupun the ultimate things that we have to understand is you can see that it will change the amino acid as a whole. Kan? Let's say this should be amino acid that we wanted in order to produce what I call it, beta hexoaminidase A enzyme. So this is a polypeptide kan? So a polypeptide can be modified into enzyme, hormone and whatnot. So for hexoaminidase, they need amino acid methionine, GLN, valine, threonine, serine, valine. So this is the sequence. But somehow now it changed into this. Right? Change into this. So bila dia bertukar jadi ni, maknanya it is no longer code for beta hexoaminidase A. Macam tu lah. It code for something else. Macam tu. Okay? Lagi soalan? Tak ada. Okay, deletion. Now we go to deletion. Okay, so we are still discussing about gene mutation or point mutation. So ingat kita dah habis pasal insertion, kita dah habis pasal substitution, right? With the example of disease due to insertion, due to substitution. Okay, now we go to deletion. Okay, so deletion is loss of nucleotide pair in a gene, hilang. Alright, so it also will cause frame shift mutation. Okay, just imagine, look at my forefinger. Let's say, okay, this one, right? My ring finger ni. So this is nucleotide 1, nucleotide 2, nucleotide 3, nucleotide 4. So what happened? My ring finger is nucleotide number 3, lost. Alright, macam John Wick the third kan? Tangan dia kena potong kat sini. Okay, so what happened? You can see frame shift mutation takes place. So the fourth nucleotide is shifted to the third. Alright, so you just imagine dia akan frame shift. Yang kalau insertion, frame shift dia akan ke sini, macam tu. deletion, the frame shift also will occur, but macam ni, ke sini. Okay, alright. So that is effect of deletion. Okay, example. This is our template strand, right? This is our sequence. So what happened? Nucleotide A and its base pair deleted. Hilang hilang. So what happened? Frame shift will happen. Ni pergi sini, ni tinggi sini, ni pindah sini, ni pindah sini, ni pindah sini, ni pindah sini. Macam tu lah. Right? So frame shift mutation happen like this. Okay? Right. So now let's see what happened. Nampak tak? Right? The effect of deletion. Tadi sepatutnya A and T. Now it is deleted. So yang ni pindah sini. Dan seterusnya. Okay? So this mutant strand will be affecting the mRNA during transcription. So you can see your codon change. All right. And then it also will change the amino acid. All right. So that is the effect of deletion. Okay. Example. All right. Example, cystic fibrosis. Okay, what is cystic fibrosis? Cystic fibrosis is happen due to small deletion in a gene encoding of a protein, right? Second example is thalassemia, alpha thalassemia. So alpha thalassemia is disruption of hemoglobin production resulting in low hemoglobin level. So thalassemia is very common, kita dah dengar kan? Kita selalu dengar thalassemia, thalassemia, thalassemia. So what is thalassemia? Thalassemia is disruption of hemoglobin, meaning the cell cannot produce enough hemoglobin, so which result with low hemoglobin level. And also person with thalassemia, alpha thalassemia, has high rate of red blood cell destruction. So red blood cell dia senang matilah, senang rosak dan sebagainya, which can cause anemia, kekurangan sel darah merah. Okay, right. So, okay. Related to chapter 10, genetic. Thalassemia is governed by codominant allele. Thalassemia is governed by or controlled by codominant allele. So, a person with heterozygous, right? You can see here, right? Heterozygous, they will have mild anemia, ataupun we call as thalassemia minor. So the allele, the genotype written kata apa? Cara nak tulis itu, you can see TMTN. T stand for thalassemia, kan? Thalassemia. M is allele lah. Allele of thalassemia, M and N. N means normal, M referring to thalassemia major. So for person with thalassemia major, the allele is TM, right? So for normal individual, the allele is TN. Okay, so cuba buat crossing sekejap. Okay, ulang kaji chapter genetic. Show the cross between a parent that is, one parent is having severe thalassemia and the other parent with mild thalassemia. So, kita punya cross macam mana? A person that is severe thalassemia, TM, cross with a person with mild thalassemia, TN. Okay, gamet. Now, macam mana gamet dia apa? Ulang kaji chapter 10.
TM.
TM. TM sebab sama kan? Okey, tak apalah, kita buat je dua-dua. Kalau tulis satu pun boleh ke tak boleh?
Boleh.
Boleh. Okay, next one is this. TN. Yes, TN. Okay, so F1. Ingat jangan suka-suka awak buat pula T. Jangan weh. Okay, ingat this is not Mendel. Okay, F1. TM. Lepas tu
TN.
TM, TN. So we want to see what is the probability of the children to have thalassemia major. Okay, betul. TM, TN, and lagi satu TM, TM. Okay, betul tak apa Mina buat tu? Okay, so what is the probability of the children to be normal?
50%.
To be normal, anak yang normal. Maknanya tak ada thalassemia.
0.
0%. Maknanya kalau one of the parent having severe thalassemia major, and the other parent is mild thalassemia, so the probability for them to have normal children is 0%. Right? Sebab you can see there is no there is no TN, right? Dekat situ tak ada. What is the probability of the children to have severe thalassemia? So percentage dia adalah 50% ataupun 1/2. Similarly to the children with minor thalassemia. Thalassemia minor. So 50% juga. Okay, faham Right, look at here. The normal person, you can see they have a normal shape of the red blood cell, normal amount of oxygen carried by the hemoglobin, but for thalassemia person, you can see they have malformed red blood cell. Okay, does affect the amount of oxygen carried by the hemoglobin. All right. Okay, any questions so far? So we have finished with point mutation. Senang ke susah chapter ni? Senang je, kan? Senang je sebenarnya. Okay, alright. So kalau tak ada soalan, kita sambung. Describe the structural changes in chromosome. Okay, structural changes. Remember, there are two different types of chromosomal mutation. Chromosome mutation ada dua, kita dah discuss tadi. One is related to structural changes, and number two is changes in terms of number. Changes in terms of number. So we are going to discuss with this remaining time that we have. We are going to discuss about the structural changes of chromosome due to duplication, deletion, inversion, and translocation. Okay, right. Okay, so chromosomal mutation involve chromosome structure, involved in changes in terms of chromosome structure. I show you already what is chromosome structure changes looks like. And number two is changes in terms of chromosome number. Okay, so the changes are often brought to problem that occurring during meiosis or by mutation. Okay, right. So the chromosomal mutation, tak kisahlah changes in terms of chromosome structure or in terms of chromosome number will have several the effect is more serious compared to gene mutation. Kenapa chromosomal mutation effect is more severe or serious compared to point mutation or gene mutation? Sebab it involves several genes. Kalau gene mutation, dia hanya melibatkan satu gene sahaja, right? But chromosomal mutation affect various genes. So that's why the effect is more, fatal or more serious or more huge compared to gene mutation. Okay, it can result in change in quantity of genetic materials. It can also cause a rearrangement of genetic material. Sebab tu kesan dia lagi besar. Okay, right. Okay, there are 4 types of changes in chromosome structure. Remember, we are discussing about changes in chromosome structure. 4 types. Okay, duplication. Duplication means double, duplicate. Okay, ada duplicate. Inversion, deletion, and translocation. Translocation means berubah tempat. Alright, so kita akan discuss lah yang mana yang sempat. Okay, right. So as we all know, crossing over produce new allele sequence combination, but no gene loci will be lost. Okay, maknanya apa? Ada lukis aduh, thank you. Let's say this is apa tadi? Aduh, kacau ayat. So bila ada boyfriend hantar air coffee dekat atas meja, jadi berasa macam ouch. Macam tu. Okay, right. So this is crossing over. Right. So you can see in crossing over, the genetic loci will be transferred. Right, berubah aja. There is no loss of genetic locus. Locus just berpindah saja pergi dekat the other sister chromatid. Kan? Tetapi okay, for deletion and duplication, it will change the number of genetic loci. Okay, cuba tengok. This is loci, kalau ingat kan, this is locus. Different locus contain or store different gene specific for a traits. For example, this locus contain gene for eye color. This locus contain gene for hair color, for example. But if this locus is lost due to deletion, hilang kena buang. So this gene will be affected. Right? So that's why it can change the number of genetic locus. But for translocation, for as kita dah belajar sebelum ni crossing over pun, it will not change or it will not lost the locus. Locus takkan hilang, cuma locus tu berubah tempat sahaja. Okay, right. Okay, let's start with deletion. Okay, what is deletion? Deletion is deficiency in a chromosome resulting from loss of a fragment through breakage. So what happened? A fragment of chromosome lost, okay, through breakage, and this chromosome will be missing certain gene. Remember, sebab dekat locus ada mungkin lebih daripada satu gene. Okay, right. So it can affect one homologous pair or it can affect both homologous chromosome. Maksudnya apa? Let's say this is homologous chromosome, right? So it can lose one loci loss kat sini hilang, ataupun dekat homologous dia pun hilang. Okay, dia boleh affect salah satu atau kedua-duanya sekali. Okay, contoh deletion. Sekejap tak pakai. Okay, so you can see example here. Let's say our one of the homologous pair, kita ambil salah satu ni, sister chromatid ni, right? The fragment is A, B, C, D, E, F, G, H. So what happened? One fragment of locus or one fragment of the homologous, one fragment of the chromosome is lost through breakage. D ni hilang. So now what happened? You only have A, B, C, E, F, G, H. So fragment D or locus D has been lost, which affect few genes. Okay, faham That is deletion. Okay. Okay, example, again, you can see this is homologous. This is homologous chromosome. You can see fragment D of one of the homologous pair lost through breakage. Dia tak melibatkan dua-dua, dia melibatkan satu sahaja of the homologous pair. Right? So what is the effect? You can see dia akan hilang dan jadi A, B, C, E, F, G, H of this one homologous. Okay? Pair. Alright? Okay. Example of this change in chromosome structure due to deletion is Cri-du-Chat. Okay, cara nak sebut dia kena betul Cri-du-Chat syndrome. Alright. So in Malaysia we have this Cri-du-Chat. Ni Madam tunjuk kat awak ni. Awak boleh Google dia nanti. Nama adik ni. Akif Nakiuddin. Awak pergi tengok dia punya Instagram ke apa ke. Right. And this is her sister. Right. So this boy is having Cri-du-Chat syndrome, which in Malaysia tak banyak sangat. Right. So Cri-du-Chat syndrome happen due to the deletion of fragment in chromosome number 5. So this is pair of chromosome number 1, 2, 3, 4, 5. So dekat chromosome number 5 tu, one of the homologous pair lost one fragment. Alright. So then affecting to be like this. The effect is like this. Apa dia? The person will have severe disabled intellectually. They will have small head. You can see this boy, look at the head. The head is small and they have unusual facial features. Bentuk muka dia agak lain. Alright. And they have cry that sounds like the cat's meow. You can go to YouTube and search for Cri-du-Chat. Right? The baby is crying like a cat's meow. Okay, and usually die in infancy or in early childhood. So this kind of syndrome happen 1 in 100,000 birth, right? So this is Cri-du-Chat happen due to deletion of fragment chromosome number 5. Madam, kena hafal ke kromosom nombor berapa semua? Kena hafal, Dek Non. Kena hafal. Okay. Alright, next. Okay, duplication. Okay, sikit lagi duplication. Duplication is aberration in chromosome structure due to fusion with a fragment of a homologous chromosome. Such a portion is duplicated. Okay, maknanya apa? Right? Thus, the chromosome will has additional set of gene. So you just can imagine, Allah cipta kita ni dah sangat complete. Tak terlebih, tak terkurang. So you can see, if let's say the chromosome or the gene is excess, terlebih pun jadi benda lain, terkurang pun jadi benda lain. Berdouble pun jadi benda lain. Alright, so this is duplication. So you can see this segment B and C duplicated. Alright, so what you can see is A, B, C, B, C, D, E, F, G, H. So the chromosome has additional set of gene. Okay, right. So this is duplication. Okay, apa kesan dia? Alright, example, this duplication happen in Drosophila. Lalat, fruit fly, lalat buah. Okay, so you can see allele for bar I in Drosophila. Normal I for the Drosophila is kidney shape. Sepatutnya bentuk kidney shape. Macam gitu, right? But now due to duplication, you can see it become bar I. Macam tu. So this is mutant. Happen due to duplication. Okay. Right? Tengok ni. Segment of homologous chromosome. So tak semestinya dua-dua pair akan berlaku. Salah satu berlaku pun dia akan memberi kesan jugalah, right? One of the homologous pair. Okay. So segment B and C duplicated. Okay, you can see here B, C, B, C, D, E, F, G, H. Okay, right. Okay, last one, inversion. Okay, Madam akan habiskan sampai inversion Okay. Inversion is aberration or deviation change of original chromosome structure resulted from reattachment of chromosome fragment in a reverse orientation to the chromosome from which it is originated. Madam kena hafal ke dia punya definition? Tak payah hafal. Untuk you punya level dah tak ada dah soalan definition. Right? Yang penting you kena faham. Okay? Right. So inversion, inverse. Dia terbalik. Reverse lah. Macam inverse. Dia bertukar terbalik tempat macam tu. Okay? Right. So the chromosome fragment is in the reverse orientation. Okay. Example. So the effect is it will change the order of gene loci on the chromosome. And this will result in phenotypic changes, what we call as position effect. Kesan dia adalah position effect because it changed the position, inverse. Example, the sequence, the fragment loci is A, B, C, D, E, F, G, H. So due to the inversion, you can see the inverse macam ni. So dia jadi A, D, C, B. Nampak tak? Inverse. Okay, A, D, C, B, E, F, G, H. So walaupun masih lagi sama, tapi dia terbalik inverse sahaja pun dia akan bagi kesan juga. So this is what we call as position effect. Okay, right. Look at here. Okay, the fragment A, B, C, D, right? Inversion takes place. Okay, so nampak dia jadi macam gitu. Even though it just happen to one of the homologous pair of chromosome. Okay, macam itulah. Alright. Okay, is there any question? Tak sempat nak habiskan sampai translocation. Okay, ada soalan?
No, madam.
No. Okay, so far can you follow and understand?
Yes.
Okay, alright. So I guess that's all. Nak sambung translocation. Kalau ikutkan Madam nak sambung, but I believe that you have class at 10, right? So I stop for a week translocation. Okay. So okay, just a reminder, don't forget test today, right? Microscopic skill test. So at 3, semua orang jawab masa pukul 3. Okay, unless kalau ada masalah apa-apa, please let me know. Okay. Right, so kalau tak ada soalan,